I'd like to begin by acknowledging the traditional owners and custodians of the land, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past and present. It's really, for me, one of the highlights and a great pleasure to welcome Professor Si Ming Man. He obtained his PhD from the University of Cambridge, followed by a postdoc at St. Jude Children's Research Hospital in Memphis. And he actually just told me that he lived in the same complex as Elvis before he became actually famous. Si Ming is already famous, so that's the difference. Currently, Si Ming is an NHMRC R.D. Wright Career Development Fellow and Laboratory Head in the Department of Immunology and Infectious Diseases at ANU. Going through Si Ming's CV is kind of it's a really excitement eye-opener. So, I mean, the number of highest ranked publications, prizes, and fellowships he already assembled on his incredible CV is very, very impressive. And for me, and I'm actually quite a bit nervous, for me and many others in the field, he's kind of a rock star. And I met Si Ming last time, first time actually in person, last year in Sydney. And I don't know what I expected, but I kind of expected like the Michael Jordan of innate immunity. And then this incredibly friendly and humbled man stood in front of me, and that makes him, for me, even a bigger star. So it's really a great pleasure that you followed our invitation, and please, thank you very much.
Well, thank you very much, Greg, for this very, very kind introduction and for inviting me to come to the WeHi once again to give a lecture series for the postgraduate students at WeHi. And I'm very happy that you chose infection as one of the themes for this year. And so it's always a pleasure to come here and to tell you about what we do in the lab. But also, I'd like to take this opportunity to highlight some of the fantastic work done at WeHi and in Australia in general in this particular area. And so today my talk is going to be focusing on inflammasome signaling, especially in the context of bacterial infections. And so, and I know you're going to get 2 lectures in this particular series with Dr. Kate Schroeder following on from next week, which you'll hear some fantastic research from her as well. And so I think it's important to start off by just explaining what are the inflammasomes in the context of pattern recognition receptors. And now these receptors are basically innate immune sensors of pathogen-associated molecular patterns, what we call PAMs. And these can come from microbial sources, including bacteria, viruses, fungi, or protozoa. And so here is a diagram that I drew highlighting some of the PAMs which are found in bacteria. And this includes all of the structural units that make up the architecture of the microbe. Including flagellum subunits, which enable the bacteria to move, bacterial DNA or RNA, the cell wall components, LPS and lipoproteins. You have the type 3 secretion systems, which helps the bacteria to inject and release its virulence factors to modify host cell functions. You also have toxin components, which can kill cells and also enable the bacteria to compete with other species. And of course, we know that these receptors can also recognize danger signals in the form of danger-associated molecular patterns called DAMs. Now, these DAMs are normally self-derived. So if you have a cells or tissue injury or damage to the cells, you can get release of host DNA or RNA molecules, extracellular ATP release, or uric acid crystals. Now, all of these are released by damaged or dying cells, and they can equally efficiently activate innate immune receptors to drive innate immunity. Now, if you are interested in this topic, Seth from WeHi recently published a paper in Nature Reviews Immunology arguing that there could be another category of these molecular patterns called homeostasis-altering molecular patterns. And in that review, he argued that these are also self-derived, but they're distinguished by their mode of activation, in that if you have a toxin which modifies the host cells, the innate immune sensors are not detecting the toxin itself, but it's detecting any of the inactivation of the host components as a result of the toxin. So again, it's an indirect molecular pattern, and it's not a molecular pattern per se, but it's actually a homeostasis modifying molecule. So let's have a look at how pattern recognition receptors work, and here is a classical example. So when you have a pathogen approaching a cell, it is immediately sensed by membrane-bound receptors, including the toll-like receptors. So for example, LPS can be recognized by toll-like receptor 4. We know that pathogens are very clever. They can invade the cells and live in this endosomal vacuole environment called pathogen-containing vacuole. In both cases, they are both detected by membrane-bound toll-like receptors. And the point of this detection is that you induce this rapid transcriptional regulation of inflammatory cytokines and type 1 interferons and anti-microbial molecules that can help clear the infections. But we know pathogens are very clever because sometimes they can lyse this environment and escape from the pathogen-containing vacuole into the cytoplasm. And here, they are able to get access to ample nutrients for them to replicate and take over the cell. So what happened in this case? Well, the host cells are equipped with several families of cytosolic receptors that help us defend against these pathogens. And they are NOT-like receptors, the NLRs, the Aim2-like receptors, the ALRs, and the RIG-I-like receptors, the RLRs. And they're equally capable of inducing transcriptional regulation of inflammatory cytokines and type 1 interferons. But we know that certain members of these NLR and ALR families have another unique functions, and that brings us to the topic of this lecture, which is they have the ability to form an inflammasome complex. So what is the inflammasome? Well, the definition of an inflammasome is that it has to be a multi-component cytosolic complex that induces cleavage of the inflammatory cytokines IL-1 beta and IL-18, and this complex can also induce cell death in the form of pyroptosis. So here is a diagram that I drew showing you the basic architecture of an inflammasome. So you need to have an inflammasome sensor or receptor which can detect the pathogen or danger signals coming into the cell in the cytoplasm. It then recruits this adaptor protein ASC, which further recruits the cysteine protease caspase-1. And this whole complex is called the inflammasome. And of course, the inflammasome have two major functions because caspase-1 can act like a molecular scissors and cleave the immature cytokines pro-IL-1 beta and IL-18 to form its mature and bioactive form. Caspase-1 can also cleave a pore-forming protein called gasdermin D, which I'm sure you'll hear more next week from Dr. Kate Schroeder. And this pore-forming protein can punch holes on the cell membrane of the cell, inducing this lytic form of cell death called pyroptosis. And so how are these inflammasome complexes formed? Well, it is formed by having different receptors recognizing different PAMs and DAMs. And in this slide, I've highlighted some nice review articles written by Australian scientists. So if you're interested, you can have a look at some of these references, both here and at the end of the lecture in my reading list. So in this series of reviews, I've highlighted a review article from the older times to give you a historical perspective of the inflammasome to more recent and up-to-date overview of the field. And so you can appreciate where the field is progressing over this time. But the point I'm trying to make here is that you can appreciate that there is a specific sensor for a specific ligand. So for example, or one or more ligands. So for example, Aim2 can recognize double-stranded DNA. The NAPE NRRC4 sensor can recognize Salmonella. NRP1B sensor can recognize Bacillus anthracis. And then you have NRP3, which kind of it's like a global sensor of PAMs and DAM, can recognize a whole range of biomolecules, and then so on. And so I know the focus of this lecture is on infectious diseases, but I'd like to remind you that the inflammasome also has important function in regulating inflammatory diseases and cancer as well. And so what does it look like in the lab when you study inflammasome? So in this image, I just culture primary bone marrow-derived macrophages, and I transfect the DNA, the activator for Aim2, into the cell, and to activate Aim2 inflammasome assembly. So then you can fix these cells and use an antibodies against one of the components, ASC, of the inflammasome, and caspase-1. And then what you can see is that these endogenous molecules are normally diffused in the cell, right? But once they sense the DNA coming into the cells, they form this distinct puncta in the cell. Most of the time, it's one per cell in the nucleus. And this is a hallmark of this inflammasome assembly. It's quite striking. And so how do these inflammasome sensors recognize PAMs and DAMs? I think we can broadly categorize them into a mechanism of direct recognition or indirect recognition. And what do I mean by this? Is that you can have an inflammasome which directly recognizes the ligands. Again, I'm showing you an example here of Aim2, which bind directly to the DNA, like a classical receptor. And this can activate the inflammasome. But then you also have receptors like NLRP3, which recognize the PAMs and DAM indirectly. And what do I mean by that is that we have no evidence in the literature to suggest that these molecules can directly be pulled down or bound to NLRP3. And if you look at this little list here, you can appreciate how many different activators can trigger activation of NLRP3. And all of them, they don't belong to any particular class of molecules. So it could be ATP, or changes in cell volume, double-stranded RNA from viruses, a simple ion flux fluctuation in the cell, or you can have some sort of mitochondrial damage, microbes such as viruses, bacteria, parasites, all can activate NLRP3. And so you can see that it responds to a diverse number of PAMs and DAMs. So we really call this a global sensor of PAMs and DAMs in the field. And so I'm going to use these two sensors as an example to highlight how inflammasome works, including some of my own work to highlight their mechanism of activation. So let's have a look at Aim2. So Aim2 is a sensor for double-stranded DNA. It is a founding member of this Aim2-like receptor family. So in human, there are only 4 members, but in mouse, there are 13 of them. And Aim2 is comprised of a Hin200 domain and a pyrin domain. And this is normally inhibited in the cell. But once you introduce DNA into the cytoplasm, this structure opens up, exposing the Hin200 domains, which directly interact with DNA through an electrostatic interaction. So basically, this means that Aim2 can recognize DNA from any sequence. So there's no sequence specificity, as long as the DNA is around 80 base pair in length. So what happened then is that you get this exposure of the pyrin domain, which allows Aim2 to recruit the adapter protein I mentioned to you earlier, which is called ASC, because it also contains a pyrin domain. So then this allows ASC to use its CAR domain to recruit the CAR domain of caspase-1, fulfilling the basic structure of an inflammasome, of a sensor, an adapter, and the effector protein caspase-1. I'd like to point out to you, depending on the context of the disease, Aim2 doesn't always behave like an inflammasome. So our work and others have shown that Aim2 can function independently of the inflammasome in the context of colitis and colorectal tumor, where it can directly inhibit cell proliferation independently of ASC and caspase-1. But in the context of infectious diseases, there is no doubt that Aim2 can function like an inflammasome, and it responds to pathogens from all domains of life. So you have bacteria, pathogens such as Francisella tularensis, which can cause tularemia. It's a potential bioweapon. Listeria monocytogenes, which is a foodborne pathogen. And you have, of course, TB-causing agent, Mycobacterium tuberculosis. And that's not surprising because they all carry DNA. And similar for DNA viruses, it can recognize MCMV and vaccinia viruses, as well as protozoal pathogens. And we have found that Aim2 can recognize fungal pathogen as well in the form of Aspergillus fumigatus. So when I started my postdoc at St. Jude, I really have this interesting question in mind, which was during an infection in a cell, when an infectious agent goes into the cell, how come the DNA can just get outside and activate innate immune receptors? So if you consider the bacteria itself, these DNAs are sequestered within a cell wall and cell membrane, and potentially in a vacuole as well, if they are vacuolar pathogens. How do this DNA actually comes out into the cytoplasm to activate Aim2? And so that was my question that I had during my postdoctoral time. And when I look into the literature, Dr. Denise Monach's group from Stanford showed back in 2007 already that in order to activate the inflammasome, during an infection such as Francisella infection, you need to have some sort of a type 1 interferon signature that is happening, and that is critically required to allow the inflammasome to be activated. So I thought that's interesting because downstream of type 1 interferon, we know that it regulates hundreds and possibly thousands of genes, right? So what could this be that can link between type 1 interferon and exposure of the receptor to the DNA? So in 2015, we published a paper showing that downstream of this transcription factor is the expression of RF1, interferon regulatory factor, which also allows DNA to somehow come out into the cytoplasm to activate Aim2. So this was interesting to me, how this transcription can do it. So we just did a simple microarray analysis, and we found that in cells lacking RF1, they have very reduced levels of these whole families of proteins called interferon-inducible GTPases. So as the name suggests, they're interferon-inducible, and they include the guanylate-binding proteins, short for GPPs, and then they also contain these members called immunity-related GTPases, or IRGs. And when I look up the literature, they have these robust anti-microbial activities, right? So they punch the microbes. So here's a phylogenetic wheel of all these members. There's so many of them. The one in blue are the IRG members. The one in red are the GBP members. So for a whole screening process, we then identified that one members in this subfamily, IRGB number 10, and two members in the GBP family, called GBP 2 and 5, were involved in driving activation of the Aim2 inflammasome. So just to show you that if we knock these genes out and create a knockout mouse, we can generate primary cells from these mice and then infect them with either Francisella or E. coli. So normally what happens is that you get robust inflammasome activation signified by very high levels of the cytokine IL-1 beta and IL-18 in the wild-type cells. But if you lack these molecules, IgB10 or GBP2 and 5, you get significant reduction of IL-1 beta. And you get further reduction if you take out both of them. And of course, we can confirm this by measuring the other cytokines, IL-18, but also measuring the level of pyroptosis by looking at LDH release from the cells. And in all of these cases, if you lack these important molecules, they have an impaired response to these bacterial agents, suggesting that these proteins are critical in regulating activation of the Aim2 inflammasome in the cells. So how do they actually work? We thought it might be interesting to use a microscopy approach to stain for these proteins. So normally they are evenly distributed in the cytoplasm of the cell. So this is a primary macrophagist. And if we add any bacterial agent like an E. coli or Francisella, and we can stain the E. coli in green, right, shown here. And then we can also stain endogenous level of these proteins, IgB10. And what you appreciate is that immediately you can see that these proteins redistribute to co-localize with these bacterial agents, looking as if they're attacking it. And we know that this is specific because it's not found in the knockout. And if you knock out its neighbor, the GBPs that work with the IgB10, the IgB10 have an impaired ability to now find the bacterial agent, and they remain diffused in the cytoplasm. And of course, we can confirm this with another pathogen, Francisella. You get not clear localization, but in the absence of GBPs, these proteins fail to localize to the bacterial pathogen. So therefore, we think that these kind of interferon-inducible proteins are recruiting to the bacteria, but how do they then mediate liberation of the DNA in the cytoplasm? So let's have a look at the cell wall of these bacteria. So for E. coli and Francisella, they're classified as gram-negative bacteria, which means that they have a grassy layer of LPS on the outside of the cell wall, surrounding an outer membrane, a thin layer of peptidoglycan, and an inner membrane here, which is different from gram-positive bacteria, which have a thick cell wall comprised of peptidoglycan. So we thought, well, in order to image such fine details, so let's have a look at using super resolution microscopy and see whether we can see anything. So to my surprise, we can actually see very clear details just by using super resolution microscopy. So here we stained E. coli LPS in green. And as you can see, it is this nice outer layer of E. coli LPS. But we also find that the endogenous IgB10 are now diffused inside the bacteria, forming this inner layer of IgB10 surrounded by this green layer of LPS. And these two images are just orthogonal views cut through the bacteria, highlighting to you that this LPS layer is surrounding an inner layer of IgB10. And this is a 3D reconstruction of this particular bacterium. We can look at further examples of this in the cell, and you can see that the IgB10 proteins eventually diffuse inside the E. coli cell cytoplasm and kind of inducing kind of this pore formation in the bacteria. You can also see examples of bacteria losing its intactness and cell shape and a loss of structural integrity as well. So we think that these proteins can localize internal to the bacterial LPS layer and induce some sort of disruption to the structural integrity of the bacteria. So then we wanted to go further and actually use transmission electron microscopy to actually see where they are exactly localized. And we can actually visualize these IgB10 proteins or GPP proteins by just using an antibodies against it, but by using a secondary antibodies containing a gold particle so we can see under EM. So this is a section of a primary macrophage infected with E. coli. So in this same image, you see two different bacterial cells in the cytoplasm. So the one on our left is a healthy-looking E. coli with a fully intact cell membrane, right? So pretty healthy. The one on our right is a kind of disrupted E. coli, kind of losing its cell membrane, but with a lot of these dark puncta, which are labeled them as IgB10. So they're specific for IgB10, all inside the bacteria. So suggesting that these molecules can go inside the bacteria and causing disruption. You can find more examples of this under EM. So in these cases, in these dark arrows, you can see these IgB10 proteins are diffusing and attacking the cell membrane of the E. coli. And whenever they're present, you see this loss of the cell membrane of the E. coli. And in regions where they were relatively free of these IgB10 molecules, indicated by the white arrows, you can see that the cell membrane remained relatively intact compared to regions which have been in touch with IgB10. I've also seen examples of where this fusion of bubbles happening. So sometimes you can see this fusion on the bacterial cell membrane that carries these IgB10 proteins, right? So this another more examples here, this kind of blob-like structure. I don't know what those are, but I thought it would be interesting to highlight those. Maybe that these IgB10 or other interferon-inducible proteins are packaged in a vesicle kind of structure and are delivered onto the bacterial surface as a lethal hit. Maybe it's more effective that way to lyse the bacterial cell. Alternatively, it is possible that the bacteria are shedding off its membrane to fend off against these anti-microbial proteins. And so this was quite interesting. So hopefully in the future we'll investigate which mechanism is these proteins operating. Is it coming in, or is it the bacteria defending against itself? So you might ask, oh, that's great. You can show in vitro that these proteins are recruited to bacteria, and we also show that recombinant version of these proteins can completely lyse the bacteria in the test tube. But what is the physiological relevance of this pathway? So to address this, we infected wild-type mice or mice lacking IgB10, GBPs, or the inflammasome sensor Aim2, or caspase-1, the effector proteins, right? And just monitor their survival over time. So you can see that wild-type mice infected with Francisella were relatively resistant to the infection, and they survived past day 12. Whereas mice lacking these interferon-inducible GTPases or lacking the Aim2 inflammasome succumbed to the infection relatively rapidly, or within 8 days of the infection, suggesting that these molecules are protective. And you can see bacterial counts as well in the liver and spleen. Both the IgB10 knockout mice or the Aim2 knockout mice are highly susceptible to the infection and harbor more bacteria in the organs compared to wild-type mice. And to link it to inflammasome, we can measure the inflammasome correlates IL-18 in the serum of these mice. And you can see that mice lacking interferon-inducible GTPases, Aim2, inflammasome, caspase-1, all have lower levels of these cytokines compared to the wild-type mice. So these data suggest to us that the IgB10 and GBP knockout mice are very susceptible to the infection, and they have an impaired ability to activate the inflammasome. So from this work, we proposed the following mechanism of activation for the Aim2 inflammasome in response to bacterial infection, which is if you encounter a bacterial pathogen such as Francisella, it can invade the cell and reside in the vacuole for a brief amount of time before it escapes into the cytoplasm. And somehow during this process, we get this expression of type 1 interferon by the cell. And these type 1 interferons can act in an autocrine or paracrine manner to then engage the type 1 interferon signaling pathway in the cell. And these can then induce a cascade of responses, including expression of the transcription factor IRF1. And these could then lead to the expression of a whole family of interferon-inducible GTPases, and of which we identify GBP2, GBP5, and IgB10 that can directly target and attack the bacteria in the cytoplasm. And so these have two major consequences. The first is that it can directly kill the pathogen, so to help the cell clear the pathogen. And secondly, it can liberate previously hidden or sequestered microbial ligands, such as DNA, into the cytoplasm, so that now these previously hidden ligands are then exposed to innate immune sensors, such as Aim2, that can recognize the DNA and induce inflammasome activation to drive further inflammation, recruitment of immune cells, and host protection. And I'd like to highlight that in addition to our work, Peter Bross's work, he's from Switzerland, also independently identified GBP2, 5 in this same pathway, and we published back-to-back in 2015. So while Aim2 is highly specific to double-stranded DNA, what about NLRP3? Because we consider this as a global sensor of PAMs and DAM, which means that it doesn't directly recognize the ligand or bound to the ligand. Instead, it recognizes some sort of physiological change in the cell caused by PAMs and DAM, rather than a specific ligand. So we know that regulation of NLRP3 is far more complex than Aim2, because it first requires some sort of priming signals. So normally the level of NLRP3 protein is very low in the cell. So you need to have some sort of boosting signals, such as a toll-like receptor signals to activate NR-kappa B to upregulate more of these NLRP3 proteins. And in the field, we call this a priming signal or signal 1. And then the cell has to then experience a second signal, which is called the activating signal, and this can be anything in the context of NLRP3. It could be ion fluxes, phagosomal rupture, or some sort of mitochondrial damage. So similar to that list I showed you earlier. So it responds to a whole range of PAMs and DAMs. And so again, just to remind you, PAMs and DAM activating NLRP3, and I'd like to highlight to you some of the key advances in the field in this area. And so one of the first studies to show that NLRP3 can form an inflammasome is by the group of Jörg in Switzerland, showing that in an overexpression system, the NLRP3 can assemble an inflammasome. And then 3 other groups, including the group of Jörg, Vishwa Dixit, and Gabriel Nunes, made the knockout mice for NLRP3 and showed that they failed to respond to a whole range of PAMs and DAMs, including ATP, bacterial RNA, or some sort of particulate matters or toxins. And so I guess now, since then, the field has really exploded just on this single inflammasome. And you'll find more than 5,000 papers just in the published literature describing the biological importance of this one single inflammasome in health and disease. And I guess I'll take the opportunity to highlight some of the fantastic work done by Australian researchers. Seth here at WeHi showed that amyloid aggregate can activate the NLRP3 inflammasome in the context of type 2 diabetes. And James Vince showed that this NLRP3 activation is highly complicated and can be inhibited by this inhibitor of apoptosis proteins in a RIPK3 dependent manner, and that this complex sometimes also require other caspases to go into the inflammasome, such as caspase 8. And so after that, in 2011, we realized that there are different ways in which we can activate the NLRP3 inflammasome. And this was discovered by Vishwa Dixit's group, and he called this a non-canonical activation of NLRP3, which helps it to distinguish from the canonical pathway. And what he meant by non-canonical is that this pathway is specifically activated by gram-negative bacteria. So in this pathway, they show that you need some sort of a priming signal first, which is a TLR4, recognizing LPS, and this helps the cells to induce caspase 11 expression in the cell. Okay, so there are some very nice work done here to show that this is the case. And then also the LPS from the gram-negative bacteria has to also go into the cytoplasm, right, in order to activate cytosolic inflammasome sensors. So, and the LPS can do that through a variety of mechanisms, including using the outer membrane vesicle of bacteria to deliver LPS into the cytoplasm. And so once the LPS go into the cytoplasm and you have very high level of caspase 1 expression, caspase 11 can then directly bind to LPS, acting as if it's a sensor. It can sense LPS directly. And then this was described by Feng Xiao's work in China. And then activation of caspase 11 allows it to oligomerize and then go on to cleave this pore-forming protein called gasdermin D. And this pore-forming protein can then punch holes on the membrane to induce pyroptosis, but also can induce activation of the NLRP3 inflammasome. So you can see there's a lot of high-profile paper characterizing this particular pathway. And of course, because of its importance in both infection and non-infectious diseases, we've now identified, work from Luke O'Neill's lab, identify that you can use small molecule inhibitors to inhibit the NLRP3 inflammasome. And of course, Dr. Kay Schroders will probably tell you about how these small molecules work. They just published a paper very recently on how this molecule, MCC950, can inhibit NLRP3. And so in the lab, how do we actually go about identifying these new microbial activators of the NLRP3 inflammasome? So one of the simple systems we can use in the lab is culture primary bone marrow-derived macrophages, see them, and then you can add any stimulation you like, toxin, bacteria, and then one of the readouts that you can use is to do a Western blotting for caspase 1 cleavage. You can measure IL-1, IL-18 secretion, and you can also observe cell death in the form of pyroptosis. And that will tell you whether your activator is potentially activating an inflammasome. So PhD students come to my lab and she's excited to potentially explore new microbial activators of the inflammasome. So what Anya Kriti did was she added a panel of clinically important bacterial pathogens into primary bone marrow-derived macrophages and just look for caspase 1 cleavage. So here you have this inactive zymogen caspase 1, at 45 kilodalton. And when the caspase 1s get activated, you get this P20 band indicating inflammasome activation. And this was not surprising because these bacteria carry all sorts of PAMs and DAM. So to make our life easier, we thought it would be interesting to filter sterilize this supernatant and just get rid of the bacteria. And what's left over from the overnight supernatant is whatever they secreted. And so using the overnight broth, cell culture broth, we then add it to the cells and see what happened. So in this experiment, the only pathogen that retained its ability to activate the inflammasome is this pathogen called Bacillus cereus, suggesting that it can secrete some sort of secretor factor to activate the inflammasome. And we have now shown that this secretor factor can indeed activate NLRP3. So if you use SCM to look at the cell morphology, this is a healthy-looking macrophage under SCM. As soon as you put the soup onto the cells, they kind of burst open with the membrane being ruptured and with the nucleus trapped inside, which is a characteristic of pyroptosis. You can see examples of the cells capturing this pill-shaped Bacillus cereus. Eventually, they undergo pyroptosis as well, right? So they die. And what's interesting is that when they die, they actually spit out the bacteria, as if they are like expelling them. And this observation is actually quite consistent with Dr. Ed Mao's work from UNC Chapel Hill, showing that pyroptosis could be an effective innate immune mechanism to get rid of intracellular bacteria so that they're no longer hiding, so that complements and other innate immune mechanisms can now get access to these bacteria and try to kill them. And so in our line of work, we then show, of course, that wild-type cells can induce inflammasome activation in response to the bug, but cells lacking NLRP3 fail to respond to Bacillus cereus. Of course, we try a whole panel of knockout cells, and it's only specific to NLRP3 and not Aim2 or NLRP1 or NLRC4. So then we did a whole screening experiments to look at clinical isolates and environmental isolates to look at what the activator might be. And we found that in 10% of the strains, all of those strains cannot activate the inflammasome, but they also lack this toxin called hemolysin BL or HBL. So indeed, we then generated an isogenic mutant lacking this toxin HBL and add this to our cells, and of course, there's no activation, suggesting that this toxin can potentially activate NLRP3 in the cell. We then go on to confirm our findings, and we use a supernatant from these bacteria. We either treat it with a control serum or use a neutralizing antibody against the toxin. So in the normal soup, you get activation, but when you neutralize the toxin in the soup, it can no longer induce activation of the inflammasome. So finally, to show that this is the real activator, we generated the recombinant proteins and add this protein directly to the wild-type cells or knockout cells. And you can see the wild-type cells can induce inflammasome activation, but not the knockout cells. So these results collectively suggest that this HBL toxin from Bacillus cereus is a novel and bona fide activator of the NLRP3 inflammasome. And so what's interesting about this toxin is that it is comprised of 3 subunits, B, L1, and L2. And I was wondering, why do you need 3 components to actually activate the immune system? So we thought there must be some sort of weird binding order happening. So to test this hypothesis, we decided to add one of the components first to the cells, and then we washed the cells extensively to remove any unbound toxin. So if they're not bound, they get washed away. So here, we put B first, then we wash the cells, and then we add the other 2 components into the cells. In this case, we get this robust activation of the inflammasome. Interestingly, if we add L1 first, this component, then wash, and then add the other 2 components, B and L2, we get no activation. Similarly, if we add L2, wash, and then put the other 2, again we get no activation. So these results clearly suggested to us that this subunit called B subunit must be the apical subunit that drives this whole process from happening. So if B is first, then who is second and who's third, or does it matter? So to do this, we then add B first, then we wash, we then add L1, and then we wash, and then we add L2. And in this case, we get this robust activation of the inflammasome, but not when we add B, L2, and then L1. So as you can see, there's a highly specific and linear order of inflammasome assembly that is required to fully engage NLRP3 inflammasome activation. And so if we look into the literature, the paper suggests that HBL can be potentially a pore-forming toxin, but no one has visualized a pore. So to do this, we generated a synthetic liposomes which mimic the mammalian membrane, and we can capture the dye inside. And the hypothesis was, if the toxin is a true bona fide toxin, it would kind of punch the holes in the membrane, and then the dye will leak out. And then we can layer at the bottom a dye-capturing resin at the bottom to capture this dye, so it turns into a different color. So using this assay, we can see that in the control condition, the liposomes are intact and no dye get leaked out and get captured. If we sonicate the liposome to rupture them, they get released and all goes into the dye-capturing resin. If we put individual components into the liposome, nothing happens. If we hit inactivated toxin, nothing happens. Only when you put all 3 components together and put it into the lysosome, liposome, you get this release of the dye into the resin. And you can quantify that here. We also submitted this liposome to cryo-EM, and you can see that in untreated liposome, the membrane is relatively intact, whereas once you add the toxin, we see this pore-like structure on the surface membrane of this liposome at 35% of the cases, suggesting that these are bona fide pore-forming toxins. And so the model that we propose for this work is that Bacillus cereus can release a tripartite toxin composed of B, L1, and L2, and these components assemble on the cell membrane in a highly sequential and linear order, defined by B, followed by L1 and L2, forming a pore which can induce ion efflux in the cell, which I didn't show you. In this case, it's potassium efflux. And so that can then induce activation of the NLRP3 inflammasome. We also tried to transfer individual components or two of the components into the cell, but nothing happened in our hands, suggesting that it's not a direct interaction between the receptor and the toxin components. And that if you inhibit phagocytosis of the toxin going inside, it also didn't do anything, suggesting that it has to be an indirect mechanism of inflammasome activation. And of course, you get robust release of IL-1 and IL-18 through the gastrostomy D pore. So the physiological relevance in this case is if we use an IP model of infection of Bacillus cereus, wild-type mice succumb to the infection very rapidly within 20 hours, whereas the inflammasome knockout mice, so lacking NLRP3 or the caspase-1-11, these mice are actually highly protected in this IP model, which is different, right? If you remember from the Francisella model, where inflammasome knockout mice are susceptible, in this case, they are resistant to the infection. To show that the HBl toxin have a role, the wild-type mice were infected with the wild-type bacteria or its isogenic mutant lacking HBl. And in this case, the wild-type mice infected with the wild-type bug succumb quickly, whereas the mutant bug, wild-type mice infected with the mutant bacteria can persist longer and survive past over a week, and they recover. And remember, I showed you earlier a nice paper from Dr. Luke O'Neill's lab and Kate Schroeder's lab showing that MCC950 is a highly selective small molecule inhibitor of NLRP3. And so we took advantage of this and injected our mice with a vehicle control, PBS, or MCC950. Just 2 single doses of the injection can completely prevent Bacillus cereus lethality in 100% of the mice. So this was interesting. So these results basically told us that cytosolic sensing of HBl by NLRP3 sensor can induce an effective innate immune response to Bacillus cereus infection. And perhaps we can even take it further and argue that therapeutic modulation of this inflammasome pathway could potentially be used against deadly bacterial infections. So hopefully I have convinced you that inflammasome can play a detrimental role or protective role depending on the context of the infection. And then inflammasome can respond directly to a specific ligand or indirectly to some sort of ion flux in the cell. So I guess for the students in the audience, hopefully after this lecture, you are able to explain what is an inflammasome and that how the inflammasome can form depending on the receptor that gets activated in the cell. Hopefully you appreciate that the inflammasome can respond to a whole range of PAMs and DAM. And then hopefully you're aware of the mechanism of inflammasome activation in response to bacterial infection. And so I've leave you with a further reading list, again, highlighting some of the fantastic work done by Australian researchers and the reviews they wrote, so you have an idea of where the fields are going or potentially consider coming into the inflammasome field. And with that, I'd like to thank you once again for inviting me. It's always a pleasure to come to the WeHi, and thank you very much for your support.
I think this is why Si Ming is a rock star. Questions? There must be plenty of questions. Yeah.
Other than caspase 1 and 11, are there any other caspases that can be activated?
So in mouse, it's mainly caspase 1 and 11 that form the inflammasome. In human, human doesn't have caspase 11. So instead we have caspase 4 and 5. And so during inflammasome activation, we get activation of 1, 4, and 5 in human cells. This one there.
With the caspase 1 knockouts, would you expect any other cell death pathways to be activated? Because it wasn't really seen in that.
Yeah, so that's a great question. Work by James Vince and others in the field have now shown that if you don't have caspase-1, the cell can eventually die because there's so much functional redundancy, right, in the cell. So what happened in that case is that apoptotic caspases come in. You get sometimes caspase 3 and 7 activation. So eventually in caspase-1 knockout cells, they will undergo some sort of apoptotic cell death. But that's a great question, yeah. So there's definitely crosstalk between pyroptosis and apoptosis, and now we have others have shown that necroptosis can come into play. So there's this whole intersection of cell death pathway that can happen when you look further down the line. This question there.
So any idea on how the GTPase is actually permeabilizing the bacteria? Is it sufficient, or does it recruit other things?
Yeah, so that's a very difficult question to answer because they don't work in isolation in the cell. So we already shown that you need at least 3 of them to work functionally, right? Because if you just lack GBP2 or 5 or IgB10, the pathogens can survive longer. So the current thinking in the field is that they are recruited in a hierarchical order. So, for example, in Inflammasome, GBP1, 2, and 5 comes in, and then they draw in IgB10. We don't know why it needs to be in such a specific order. And then after they all recruited, how do they then lyse, right? So if you look at the structure of the protein, they do have these transmembrane regions. And in the lab, we did try to chop that region out and synthesize them directly and just incubate that with the bacteria, just in the test tube. So the transmembrane region of this interferon-inducible GTPase seems to be essential in driving membrane damage and killing.
Is there a specific pathway that's inducing the type 1 interferons for the GTPases?
Yes, so what I didn't show you is that when the bacteria first escape the vacuole, it has to be sent somehow in order to induce type 1 interferon, right? And so it depends on the pathogen. So for gram-negative bacteria, most of them, it goes through the TLR4 pathway because it can go through MyD88 and TRIF. So the TRIF pathway goes to upregulate interferon. For Francisella, it escapes and activates the DNA receptor called cGAS and STING. So in that pathway, they're also equally capable of inducing type 1 interferon. So there are several ways at the beginning how interferon can be induced to then activate the whole type 1 interferon signaling cascade.
Following Grant's me.
Yeah, sorry.
Following Grant's questions, do you know those GTPase proteins needs to be in GTP bound form or in GDP bound form?
They can bind to GTP or GDP. And in order for them to be activated, the globular domain has to bind to the GDP.
And have you identified the upstream regulator like the GTPase activated proteins or the guanine nucleotide exchange factors?
Yeah, no, we haven't. Yeah, to-do list.
So that data you showed at the end, and you rescued the mice with the MCC 950, but presumably there's still mice that are alive with a severe bacterial infection. So what sort of happens? Did you look at what happens after that, or if other innate pathways sort of take over?
Yeah. We were surprised that it was so effective as well, because if you look at the inflammasome knockout mice, not 100% survive, right? But when you treat it with MCC, my student was telling me they look completely healthy. They were running around, looking as if they're not even infected. So in this model that we use, what's interesting is that the bacteria are not chronic, so they're not persistent. So the bacteria, so the mice either clear it or they succumb to the infection. So after day 2, all the bacteria is gone already from the system.
I was just wondering with the activation of the NLRP3 inflammasome that's so detrimental, are there other regulatory mechanisms that are in place to sort of prevent that from happening? What is so specific about this infection?
So in general, there are so many mechanisms by which NLRP3 is regulated. So the simplest one is that what I described to you as priming signal. So normally it's expressed in very low level. So Which is good, right? So when the cell sees something, it just doesn't just explode and activate NLP3 because so many things can activate it. So it needs to have a signal 1 first. So if a bacteria is coming in, it needs to have some sort of LPS, some sort of other PAMs to then induce sufficient amount of NLP3. And then another major pathway to regulate the NLP3 inflammasome is autophagy. So autophagy can get rid of inflammasome once it's signaling. So then you don't have this persistent inflammasome occurring in the cell. So that is another mechanism by which inflammation can be suppressed.
This two-step control mechanism seemed to be a common feature of both parts of your talk. So I can see the rationale for it in the second case, but why are those GTPases, why do they have to be interferon inducible? Wouldn't you want them around all the time?
Yeah, no, the immune system is fascinating why you need this two-step mechanism. What I'm thinking is normally during homeostasis, you actually don't want these interferon-inducible GTPases to be floating around because their mechanism of action is targeting membrane. So imagine if you have abundant of these and you're getting mislocalization onto the plasma membrane or the mitochondrial membrane or the ER or the nucleus, then your cells can be severely stressed, right? Only when you have an infection, then these GTPases can come in. So, okay, if they mislocalize to some host membrane, that's fine, because eventually you want to get rid of the pathogen. So I think that is an essential consideration. You don't want them to be around to just damage host membrane. That's one possibility. The other possibility is that you have interferon loop first, and then you get these GTPases, interferon-inducible GTPases in the cell before the Aim2 inflammasome activation. So you really want these proteins to help you clear the pathogen first. And if they are successful, then you don't need the second pathway of inflammasome activation, right? So if there's only a few bacteria, you might be able to clear it up and kill the bacteria. And so there's not much DNA floating around to activate Aim2. But if the bacteria is so stealth and it can just replicate and take over the cell, in that case, maybe it's better to be sacrificial and just lyse everything, and then the DNA comes out and you get this inflammatory form of cell death. So I think it's better to just control the infection first before you go into this cell death inflammatory pathway.
Just wondering if you see much variation in that linker protein between the sensor and the caspase. Is that the same across all the inflammasomes, or is it different?
Yeah, so the linker protein you're talking about is the ASC protein. Yeah, so that is a very unique adapter. It is used by most inflammasomes, except a few. So some inflammasome sensors carry a CAR domain instead of a parent domain. So that means it can directly interact with the CAR domain of caspase-1. So you can have sensor caspase-1 directly. You don't need this center linker bit in it. And in those cases, you also don't see those nice specks that I showed you in the picture. They don't form these giant complexes. Only in cases where you have this linker protein, you see this really nice speck. But yeah, it's a fascinating biological question. We don't know what's the difference between having it and not having it, and why you need this giant complex versus inflammasome complex you can't necessarily see under the microscope, a smaller one. And is there a difference in the dynamic or the magnitude of inflammation? We don't know.
Your HBL port, you indicated that potassium is on the substrate. Is there any other substrate for that port, or is it just the potassium efflux which then actually produces?
Yeah, that's a great question, because why potassium, right? Because the pore is actually non-selective. So, but we did try and control by adding sodium instead of potassium on the outside, right, to block efflux, and it didn't do anything. So the cells still can die. So something about potassium is inhibiting the NLP3 inflammasome if you have excess of it outside, but not sodium. So I don't know where the specificity comes from. Presumably, most iron can go across the toxin pore, but we don't know why. But that's a great question.
Any other questions? So there will be a student lunch with at 1 o'clock. I think there is actually the Department of Infection Immunity, so we have to finish up on time. But there will be lunch with Si Ming Man in the boardroom, so students please come along. Si Ming is here for another couple of hours, so and that leaves me to thank Si Ming for a great presentation. And,